Our chosen paper is entitled "Distribution and Role of Peripheral Arterial Chemoreceptors in Cardiorespiratory Control of the South American Rattlesnake Crotalus dorisus." In the introduction, a primary role of the cardiorespiratory system is to supply oxygen to meet the metabolic demands of tissues. To achieve this, peripheral arterial chemoreceptors constantly monitor blood oxygen levels and help adjust breathing and circulation to match oxygen supply with tissue demand. These chemoreceptors are found in all vertebrates studied so far and are closely linked with pharyngeal arc derivatives. However, in reptiles, functional peripheral chemoreceptors have only been confirmed in turtles. For other than reptiles like lizards, snakes, and crocodilians, evidence is either limited or entirely lacking. Because reptiles can alter blood flow using cardiac channels, both the respiratory and cardiovascular system plays a key role in regulating oxygen. Given these complexities, our study aims to first locate functional chemosensory areas in rattlesnakes, second, determine the reflex response to hypoxemia, and third, identify the oxygen sensing cells based on the neurochemical markers. Ultimately, we seek to understand how peripheral chemoreceptors regulate both breathing and blood flow in reptiles and how these systems have evolved across vertebrates.
For this study, South American rattlesnake were obtained from the Butantan Institute in Sao Paulo and transported to Universidad Estadual Paulista. The snakes were housed in a vivarium at room temperature 28 degrees Celsius under natural photoperiod. They were first sedated using carbon dioxide, then intubated and ventilated artificially at a rate of 6 breaths per minute with a tidal volume of 25 milliliters per kilogram. Anesthesia was maintained using 0.5 to 1% isoflurane. A ventral surgical incision was made just in front of the heart to allow for the placement of transonic flow probes around the pulmonary artery and left aorta, which enabled measurement of pulmonary and systemic blood flow, respectively. Additionally, arterial catheters were inserted into the pulmonary and caudal arteries to monitor pulse pressure in the pulmonary and systemic circulations. To assess ventilation indirectly, needed electrodes were placed under the skin for impedance measurements. The researchers divided the snakes into two experimental groups to test different chemosensory sites. Group 1 included snakes that had catheters inserted into the aortic arc and pulmonary artery to locally inject sodium cyanide and test if these areas functioned as chemoreceptors. For Group 2, they had a catheter placed into the carotid artery aimed at the carotid bifurcation, another suspected chemoreceptor site. The purpose of dividing the snakes like this was to allow site-specific stimulation, meaning they could test how each individual area affecting breathing and cardiovascular responses when exposed to a hypoxia-mimicking chemical. This setup helped them determine the specific roles and reflexes of each chemoreceptive region in the snake's body. So as we can see here, the denervation was done to confirm that sodium cyanide induced responses were due to peripheral chemoreceptor activation. The researchers surgically cut specific vagus nerve branches. To the group 1, snakes had the nerves to the aortic arc and pulmonary artery severed, while group 2 had those to the carotid bifurcation cut along with the nearby vascularized gland. So the main vagus trunk remained intact. For testing, sodium cyanide, a chemical that mimics low oxygen, was injected in a small doses, 0.5 mg in 0.1 ml, into targeted blood vessels using catheters. The saline was flushed afterwards, and control injections with saline alone ensured the responses were specific to sodium cyanide. The injections were randomized and spaced to avoid overlap in effects. So here, the data acquisition and statistical analysis were used to track changes in heart rate, blood flow, blood pressure, and ventilation before and after sodium cyanide injections. These values were recorded using WINDAQ software and analyzed over set time intervals. Calculations like cardiac output, stroke volume, systemic resistance, and total ventilation help determine how each chemoreceptor site influenced cardiorespiratory function. Statistical tests, such as ANOVA and t-test, were applied to check if the changes were significant. The Holm-Schedach test was specifically used after ANOVA to identify which time points or conditions had real differences, among minimizing the chance of false positives from multiple comparisons.
After the thorough discussion of the methodology, we will now analyze the results and discussion. This figure shows the schematic and actual anatomy of the three hypothesized chemoreceptor areas: the carotid bifurcation, the base of the aorta, and the pulmonary artery. The black arrows that we can see here highlight the branches of the vagus and glossopharyngeal nerves that innervate each chemoreceptor. As described in the methodology, NACN was injected to stimulate the chemoreceptors. Following NACN administration, systemic blood flow, pulmonary blood flow, and heart rate were recorded. Based on the figure, we can observe that systemic blood flow decreased across all three hypothesized chemosensory areas. Pulmonary blood flow increased in the carotid bifurcation and aortic arc. In the pulmonary artery, the pulmonary blood flow initially decreased at 120 seconds but later increased and returned to pre-injection levels at 240 seconds. As for the heart rate, it increased in all three sites compared to their pre-injection state. Now, we can deduce from the data that the decrease in systemic blood flow at all three sites suggests that vasoconstriction occurred. It is a reflex aimed at preserving oxygen delivery to vital organs, such as the brain and heart, in response to hypoxia. The increase in pulmonary blood flow implies a redistribution of blood toward the lungs to compensate for perceived oxygen deficiency, while the increase in heart rate across all groups represents a tachycardic response that helps increase cardiac output and accelerate both oxygen delivery and carbon dioxide removal. These cardiovascular changes are consistent with the chemoreflexes observed in mammals and other vertebrates. The similar responses triggered by NACN injection at each site support hypothesis 1, which posits that these regions contain functional peripheral the changes in shunt fraction, cardiac output, and systemic resistance, which can be observed in this figure, further confirm that all three sites serve as active peripheral chemosensory areas. In the carotid bifurcation, the increase in shunt fraction suggests that more blood was rerouted away from the systemic circulation, likely toward the pulmonary circuit to enhance gas exchange. The increased cardiac output and systemic resistance reflect a compensatory cardiovascular response to maintain blood pressure and oxygen delivery. In the aorta, the shunt fraction peaked and then decreased, indicating a dynamic adjustment of blood flow. The increase in systemic resistance likely indicates again vasoconstriction to preserve arterial pressure. The cardiac output decreased slightly, but the values remain close to the pre-injection levels. In the pulmonary artery, both shunt fraction and systemic resistance rose significantly at 120 seconds post-injection. In contrast, cardiac output decreased markedly, likely reflecting a stronger vagal reflex that reduces heart pumping to limit oxygen demand after hypoxic conditions. These findings further confirm that each site can independently trigger cardiovascular reflexes via chemoreceptor activation. The timing and magnitude of responses vary, likely due to anatomical and functional differences. The table 2 shows the effects of NACN on cardiovascular variables. So we can see here the significant changes in stroke volume in aorta and pulmonary artery, whereas no significant change was noted on the carotid bifurcation. There was also noticeable change in the mean arterial pressure in the pulmonary artery group, while no significant changes were observed in the carotid bifurcation and aorta. But systemic pulse pressure showed significant changes in the carotid bifurcation and aorta, especially in the pulmonary artery, where the changes were most pronounced. To determine the statistical significance, the Home-SIDAC post-hoc test was used following ANOVA. This method adjusts for multiple comparisons while maintaining statistical power. It helped identify which specific time points and vascular sites showed significant differences from their pre-injection baselines. So the significant changes observed across all three vascular sites support the hypothesis that each one contains functional peripheral chemoreceptors. However, again, the strength and the type of response varied by site. Among them, the pulmonary artery exhibited the most dramatic changes, particularly in the systemic pulse pressure, emphasizing its high chemoreceptor. This figure shows the original traces of NACN injection effects at the three sites. On the left side, we can see here the hemodynamic variables compared between intact and denervated sites post-injection. On the right side, we can see ventilation traces. A visible ventilatory response is observed only when the sites were intact, confirming that the carotid bifurcation, aorta, and pulmonary artery mediate the response to NACN. It is evident that NACN causes increased ventilation and changes blood flow, which are abolished or diminished after denervation, further proving that these are functional chemoreceptive areas. All three regions mediate strong ventilatory responses to NACN, but the pulmonary artery group shows the greatest response, particularly in total ventilation and amplitude. The breathing frequency increased initially in all three groups following NACN administration, but then quickly decreased. In breathing amplitude, it is also increased in all three groups after NACN injection, followed by a subsequent decrease that varied across. So this table presents the numerical values of the respiratory responses to NECN of the previous figure. It clearly demonstrates the differences in response magnitude between the three sites. So in figures 4 and 5 together, they highlight the major shifts in blood flow and pressure after the NACN injection. They also demonstrate that these responses are dependent on nerve innervation, as shown in figure 4, where responses disappeared following denervation. These results further underscore the important role of the pulmonary This figure presents hematoxylin and eosin stained or H&E stained sections of the aorta. Immune labeling reveals both VAC positive cells or the green ones in the panels D and E, and serotonin positive cells or the red ones in the panels F to H. So this confirms that the aorta both contains cholinergic and serotonergic chemoreceptors. The H&E stained sections show the anatomical structure of the carotid bifurcation. Immunofluorescence images, the panels F to G reveals serotonin, immunoreactive cells, those the red ones, with DAPI nuclear staining, the blue ones. And notably, VAC labeling is absent, or the green ones that we observed from the previous figure, indicating that the carotid bifurcation contains only serotonergic but not cholinergic chemosensory. Similar to aorta, the pulmonary artery sections display VAC positive cells or the green ones that we can observe in the panels E to F and serotonin positive cells. And these findings confirms that the pulmonary artery has the third distinct chemosensory site containing both serotonergic and so the immunolabeling revealed that the carotid bifurcation is unique among the three sites in having only serotonergic oxygen sensing cells, while both the aorta and pulmonary artery have both serotonergic and cholinergic elements. That supports the hypothesis that different chemoreceptor sites have specialized roles in regulating cardiorespiratory function. And the previous figures also sums up or confirms the first hypothesis that rattlesnakes have three distinct chemoreceptor sites, specifically the South American rattlesnake, the carotid bifurcation, aortic arc base, and the pulmonary.
In our conclusion, this study examined oxygen chemoreceptors in rattlesnakes, identifying their location, function, and neurochemical characteristics. These chemoreceptors were found in pharyngeal arc derivatives, the carotid bifurcation, aorta, and pulmonary artery, matching locations seen in turtles and amphibians. The presence of multiple receptor groups sensing either arterial or mixed venous blood may benefit animals with intracardiac shunts. We confirmed that NAC stimulation mimicking anosmia triggered ventilation increases and cardiac shunt adjustments across all sites. This finding suggests that the blood flow regulation plays a key role in maintaining arterial gas levels. Through hypoxia is rare, hypoxemia is common due to their undivided ventricle, making shunt regulation more vital than ventilation. In low oxygen, rattlesnakes increase ventilation, reduce right to left shunt, and raise heart rate to preserve blood gas homeostasis. Finally, the presence of acetylcholine and serotonin in chemoreceptor cells implies their role in O2 sensing with similarities to chemoreceptors in In our conclusion, this study examined oxygen chemoreceptors in rattlesnakes, identifying their location, function, and neurochemical characteristics. These chemoreceptors were found in pharyngeal arc derivatives, the carotid bifurcation, aorta, and pulmonary artery, matching locations seen in turtles and amphibians. The presence of multiple receptor groups sensing either arterial or mixed venous blood may benefit animals with intracardiac shunts. We confirmed that NAC stimulation mimicking anosmia triggered ventilation increases and cardiac. This finding suggests that the blood flow regulation plays a key role in maintaining arterial gas levels. Through hypoxia is rare, hypoxemia is common due to their undivided ventricle, making shunt regulation more vital than ventilation. In low oxygen, rattlesnakes increase ventilation, reduce right to left shunt, and raise heart rate to preserve blood gas homeostasis. Finally, the presence of acetylcholine and serotonin in chemoreceptor cells implies their role in O2 sensing with similarities to chemoreceptors in